Hi, can you hear me? Can you hear me?
Unmute myself.
Yeah.
That sound good? Okay. Hopefully the Zoom folks will let us know. I've turned my camera off. Okay.
Yeah, we don't need.
Okay, good. Yeah, excellent. Okay, I'm gonna mute myself for now. Yeah.
Do you want my mic or?
I've got a mic. Yeah.
Oh,
I'll set up.
okay.
Yeah. Thanks.
Do you want to use this mic? I'm fine. Yeah.
Okay.
Fine.
Okay. Cool.
Hi, everyone. Oh, and welcome the prospective students, and hope you are enjoying your visit so far. Today, I'm really pleased to introduce Dr. Amy Willis, my colleague and friend, who is an associate professor here at UW Biostatistics. And she's also the PI of the Statistical Diversity Lab. And Amy and the Stat Diversity Lab developed tools for the analysis of microbiome and biodiversity data. And Amy's definitely a leading expert in this area. And she's very passionate about statistical methodology, reproducible science, ecosystem and host health, and collaborating with scientists who share these values. And Amy is the recipient of an NIH Outstanding Investigator Award, at UW Outstanding Faculty Mentor Award, and the UW Outstanding Faculty Teaching Award, the all-star faculty. Yeah, so thanks, Amy, for the seminar, and the floor is all yours.
Thanks so much, Ting. Can everyone hear me okay? Awesome. Where are our prospective students? Hands up nice and high. Wow, welcome. Awesome. I'm so glad you're here today. Thanks so much for coming to Seattle to visit. And I really hope I get to see a lot more of you in September. So hope you're having a wonderful day, and thanks for making time for a seminar. Ting, also thank you for the lovely introduction, and thanks to Ting and Ken for having me give this seminar. So this talk really begins with the advent of high throughput sequencing. So high throughput sequencing has totally revolutionized the way that we study biology and medicine. It's revolutionized the way we study oncology. It's totally changed how we understand how cells differentiate and differentiate into different types of tissues. It's also revolutionized ecology and the way that we study organisms living in the environment. And so one of the ways in which the advent of high throughput sequencing has altered the way that we study ecology is by creating a vast intellectual distance in between what we'd like to study and what we can study. So what do I mean by that? In classical ecology, there's typically someone with a clipboard and a floppy hat sitting by a river counting frogs on a clipboard. But what's happening now that we're studying ecology with high throughput sequencing is we're blasting open cells and pulling out their DNA, potentially amplifying it, targeting specific regions of a genome, and putting it through some machine that measures which base pairs we're seeing with lasers. You can tell that I'm not someone who has a deep understanding of the engineering required to make this happen, but I see Kevin's nodding, so I'll take that as validation. And then at the end of all of that, as the machine spits out these letters, we have to deal with this post-processing stuff as well. How do we turn these letters into meaningful quantitative units that then we might go ahead and analyze as statisticians? And so you can see that there's this sort of big separation between what we'd like to study and what we can study. And the distortion that occurs in between those steps is the focus of this talk today. So the particular setting for high throughput sequencing that I'm going to focus on is the study of microbiomes. So what are microbiomes? Microbiomes are communities of microscopic organisms. And you might say, well, why should I care about them? I don't mean to plunge anyone into an existential crisis on this fine Thursday, but in fact, you're not fully comprised of your own cells. You're actually about 10 times more bacterial cells than you are human cells. And you might say, okay, well, these bacterial cells are really tiny and I'm really big, so maybe that doesn't matter. Maybe it's actually the functional ability that these bacteria have. And I would say to that, well, actually, if you're thinking about the human genome, you have about 22,000 genes, and the complement of all of the genes in your microbiome is about 8 million genes. So that's pretty incredible. They're doing a lot for you. What are they doing? This amazing paper that I read recently described them as complementing our physiological deficits, which I thought was pretty extraordinary and also kind of bold. So what is something that you can't do is digest complex plant polysaccharide. So you can't digest dietary fiber. That is your microbiome breaking down those plant cells for you, helping you derive energy from them. They're also first on the scene when a new virus appears and helping your immune system support you to not be infected by viruses. And then on days when I'm feeling sentimental, I like to remember that because the first complex eukaryotic organism was created by the engulfment of a bacterium by an archaeon, they're actually our very distant cousins as well. So we really have a whole lot to thank our microbiomes for. So as statisticians, we might ask what data structures we're typically working with when we're thinking about analyzing microbiome data. So a common microbiome data structure are these sample by category tables. So throughout this talk, I'm going to let Wij denote the number of times that some strain or category or species, or I'm going to refer to them as taxa, taxon J is observed in sample I. So in this example, we can see that Lactobacillus crispatus was observed 19 times in sample 1, and Lactobacillus innes was observed 4 times in sample 1. This is the data structure that we're typically working with in microbiome science. So when we go to think about modeling and we go to think about how do we connect this data that we have to parameters that we care about, we have to start thinking about, well, what are the environments that we'd like to study comprised of? And so I'm going to let Pij denote the true relative abundance, in some relative sense, of taxon J in sample I. And I'll note that a really common assumption that lots of methods that go about connecting types of data we saw on the previous slide to parameters make the following assumption. They assume that the expected value of these counts is proportional to that relative abundance, where that proportionality constant depends on the sample. Okay, and so this is a really important assumption, appears in all sorts of different methods. And a really natural question to ask then is, well, how can we investigate and validate this assumption? Is it a reasonable one to make? So a really incredible tool that we have in microbial ecology are called mock communities. So mock communities are fake microbiomes. They're artificially constructed to have a known composition. And they've been really widely used in computational biology, but they haven't really been used by statisticians. And I thought this was a missed opportunity. So what's a mock community? I've got 4 mock communities here. This first mock community is comprised of 100% Streptococcus agalactiae. The second mock community is comprised of a 1-to-1 mixture of Aptopia vaginae and Prevotella bivia. And this third mock community is a 1-to-1-to-1 mixture of Lactobacillus crispatus, Lactobacillus innes, and Prevotella bivia. So one thing that you may be interested to know is that data that I showed previously is actually generated from these mock communities. So I'll take a second just to let that sink in. And I'll then make 2 observations. So the first is that we're observing taxa in samples in which they're known to be absent. Okay, so we're observing 19 copies of Lactobacillus crispatus in this first mock community, which is known to only contain Streptococcus agalactiae. The other thing that I want you to notice is that despite certain taxa having equal relative abundances, we observe many more copies of some taxa than others. So let's just look at these first 2 columns, and we'll note that despite the fact that Lactobacillus crispatus and Lactobacillus innes both are present in about 33% relative abundance, we're observing about 2.5 times more copies of Lactobacillus innes compared to Lactobacillus crispatus. So I'm just going to say that again because it does become important. So despite equal mixing fractions, some taxa are observed many more times than others. And then in addition, despite purportedly being absent, some taxa are observed. So as a statistician, I would say there are at least two possible goals that we can consider based on these observations. And so the first is to say, well, given this noisy data, Wij, let's develop a method to recalibrate and estimate true proportions in a way that accounts for whatever sample to sample or taxon to taxon contamination is present. But our goal here could be to recover those true proportions. And that's something we did, but that's not what I'm going to talk about today. What I'm going to talk about today is a method to estimate meaningful differences across groups where those groups are defined by covariates. And as I go through this talk, I'm going to explain in more detail what I mean by meaningful differences. So I'll also note that all of the work that I'm going to be talking about for the rest of today was done in collaboration with one of my former PhD students, David Clausen, who was just an absolute joy to mentor. Okay, so we've talked a bit about this common working assumption and thought sort of primed you already to think about how we're going to go about validating it. And so we've seen that in these samples in which taxa are present in the same true relative abundance and the same samples, they have the same sample-specific proportionality term, we are observing some taxa many more times than others consistently. So I want us to start thinking about models that might better explain this observation. So I've pulled out 5 mock communities here from this incredible data set collected by Brooks and co-authors. And I pulled out these 5 mock communities because they both contain, all 5 of them contain the same 2 organisms, one of which is in green and one of which is in blue. And both of these organisms are present in every one of these communities in equal relative abundance. But that green taxon is observed many more times than its blue counterpart. And actually, something we noticed as we looked at this and scratched our heads was that we're observing approximately the same over-detection of this green taxon relative to this blue taxon. So we're observing about 18 times more copies of Lactobacillus innes compared to Streptococcus agalactiae, consistently across all of these samples. So that's pretty nice. I would say that's not too much noise, especially for high throughput sequencing data. So am I cherry picking this data? Am I cherry picking this pair of taxa? This pair of taxa. I've got pairs of taxa on the x-axis here, and then ratios of numbers of copies observed in these samples on the y-axis. And you can see that we observed consistently about 18 times more copies of innes compared to agalactiae here. And what happens when I show you all of the other pairs is that we're also observing these over-detection or under-detection effects. But the over-detection depends on the combination of taxa that we're thinking about. And I would say that in all of these cases, while there's some variation around their mean, generally there's a high degree of low variation around its mean, regardless of the number of other taxa present, regardless of which other taxa are present in the mixture, and in particular, how many taxa there are. Okay. So I would say this is pretty strong evidence against this first model that we talked about, where we're thinking about expected counts as just being proportional to sample-specific terms and true relative abundances, and much stronger support for a model that accounts for the taxon, this category-specific effect. Okay, so I'm making a big deal of this, and so I want to take a moment to say, why is this so important from an applied or from a modeling perspective? So let's, instead of thinking about on the count scale, let's flip to thinking about the relative abundance scale. So let's just think about dividing through our counts by the sample-specific totals across all of the categories. And now we can think of observed relative abundances as being proportional to true relative abundances in these taxon-specific detection effects. And so let's just think about a scenario where, let's just think about population data. So we've got noiseless, infinitely many samples, and we observe the following trend. We're in some, let's say, randomized controlled trial, and our goal is to decrease the relative abundance of a pathogen, which I'm going to denote in orange here. And looking at this data, we would say we've been successful. We've successfully decreased the relative abundance of this pathogen from 42% in our control samples to 24% in our treatment samples. But unfortunately, data that could have generated this observation is the following. The relative abundance of this orange taxon could have actually increased from 20% to 33%. Okay, so what could have explained this effect? Well, we could have differential detectability of these taxa in our samples. So what's going on here is that we've got one taxon denoted in green that's very easy to detect. It's 18 times easier to detect than this blue taxon, and this yellow taxon is about 6 times easier to detect than this blue taxon. And so what's happening is that the relative abundance of this green taxon is also changing. It's changing from 5% to 33%. And because it's very easy to detect, it sort of eats up a lot of the relative abundance pie that's available, edging out that orange taxon effectively. And so what we're seeing here is this phenomenon whereby the abundance of any given taxon not only depends on, or the observed relative abundance of any given taxon doesn't depend only on its relative abundance, but also the abundances of all the other taxa and how easy they are to detect. So we've got this tying together of our entire community in this sort of data structure. So I would say a fun fact, but it's actually kind of a sad fact, which is that differences in relative abundances, so if we're thinking about trying to make statements about changes from 20% to 33% in the relative abundance of these pathogens, these aren't identifiable in the absence of knowing information about how easy these taxa are to detect. And so it raises this question of, well, what parameters can we estimate about our true community from our observed data in the presence of these sample-specific detection effects? Sorry, taxon-specific detection effects, as well as sample-specific detection effects. Maybe this is a good place to pause and see if we have any questions. Clearly in lecturing mode this quarter, so very accustomed to periodically pausing to see how we're doing. Oh yeah, thanks so much, up the front.
Like, again, why can't you find given like based on like what you know from
Yeah, great question. So we're thinking about the setting here where we don't know the actual relative abundances of the taxa. So all we're observing are empirical relative abundances. And what I would say is that if we're thinking about this sort of actual column, this actual data set here, is that if, that would be consistent with the observed being the same as the actual, with all of the efficiencies all being equal. And so exactly as I think you're alluding to, in the absence of having a sort of calibration data, we're not able to estimate these detection effects, and we're not able to sort of undo that mess that happens when we multiply our observed, our true abundances by our detection effects. Does that answer your question?
sorry.
Yeah, please go ahead.
Is the detection effect the same for a string? So could you like use the walking
Great question. So you can estimate these detection effects from mock community data if you have it. Yeah, we're thinking here about the scenario where we don't have mock community data. We only have communities of unknown composition, but absolutely, in the presence of that calibration data, we can estimate those detection effects. Thanks for the question. Antonio, did you have a question as well? Excellent, cool. Does anyone else have questions? All right. Let's think about what parameters we can estimate. Thanks for the great questions, everyone. Okay. Okay, so at this point, I want us to sort of segue a little and think about some other, what would we do in our perfect world? Ask this question, what would we do in a perfect world? And so what we really care about are not relative abundances. Relative abundances are obtained by some artifact where we're deciding on what's a total and dividing through by this total. And that's not really how biology works. So what like activates the immune system is total microbial load, how much bacterial DNA or how many copies of bacterial cells are present in the environment. And so I'm going to introduce a new data structure at this point. I'm going to call it YIJ, and I'm going to think of them as some measurements of bacterial abundance on the absolute scale. And so we can measure these with some very labor-intensive, very time-intensive experiment. We have to develop category-specific primers and do amplification and perform qPCR. But this data can be collected, and I'm showing some data here from Scott McClelland, who's a faculty member in global health at UW, who's laboriously developed these this experimental protocol for 8 different taxa. Actually, there's 14 different taxa, but I'm showing 8 here. And we can observe the concentration, some absolute measure. On the y-axis, we're thinking about here number of gene copies per cell. And we're seeing there of the order of like 10 million or so. This was done in the context of an HIV case control study. So this is what's existing in the environment. And let's think about what we would like to model, or what we would like to answer if we had access to this data. So if we observe this data, then what we might do is fit a log linear model to estimate fold changes and the true abundances across groups defined by covariates. And so the question I'll say is, well, given that we don't have this Yij data, we only have this cheap and noisy and corrupted Wij data, can we still estimate beta? And the answer is sort of, and that's what we're going to spend the rest of this talk talking about. Okay, so how do we do that? So we're going to start by taking that mean model that I've heard no disagreement on, where we're thinking about expected counts as being proportional to true relative abundances with sample-specific and category-specific detection effects. And I'm going to now think about them in terms of our, I'm going to move from our relative abundance scale to our total concentration scale, so Pij to Yij. That involves some sample-specific additional constant term. I'm going to wrap that up with the Ci term, and I'm going to exponentiate that and call that term together. I'm going to call it Zi. You can call it Zi. Most people laugh, but I think everyone in this room's heard that joke from me before. So thanks, everyone. I'm also just going to, for ease of estimation later, I'm going to think about modeling these category-specific terms, these EJs, detection efficiencies, on the log scale or exponentiated scale. So I'm going to go from EJ to delta J. And so from there, if we substitute in our log linear model, we see that we have this log mean that has an additive sample term, an additive category term, and then our regression model in terms of covariates and regression parameters, which is now a matrix. So I'll just note that beta is a matrix. It has a row for every covariate, and it has a column for every taxon, every category. Okay, so now we have to think about, are the parameters of this model identifiable from the data that we have? And so you can imagine that in most scenarios, people are thinking about, I guess most generally, one being in the column space of this matrix of covariates X. And so in this case, when we've got sort of an intercept column, our delta J terms and the first row of that beta matrix are not individually identifiable. And so what we're going to do is we're just going to drop this delta J term, these detection effects from our model, sort of incorporate them or absorb them into that intercept term, and we're going to say we lose interpretation for that first row of the betas. That's the loss that happens with this identifiability. That's something we can't estimate from this data. Okay. So what about the other terms? What about these sample-specific terms and then these regression parameters? And so it turns out that if we take any p-dimensional vector and we add it to every column of that matrix of regression parameters, and then we subtract a linear combination of that same p-dimensional vector from every one of our sample-specific terms, we end up with the same mean that we would have started with originally. That's true for any p-dimensional vector. And so what that means is that we're able to identify beta to within an equivalence class. And the form of that equivalence class is as follows. It's given by constant row additions. So what this means is that we can identify the equivalence class of beta, but we can't distinguish between elements of that equivalence class. So I'll say that in a couple different ways. If you're a linear algebra person, you might think of this as not being able to estimate the levels of any category, but you are able to estimate contrasts between categories. If you're a data person, you might say, okay, well, I can't estimate fold differences, but I can estimate differences in fold differences across categories. And so in order to do estimation of these betas, we need to find a way to identify only a single member of this equivalence class. And the way that we're going to do that is by imposing some identifiability constraint, specifically an identifiability constraint on the rows of this beta matrix. Okay, so let's take a look at this visually. So let's say I had my true Yijs, sorry, my real total concentrations, my real absolute abundance data. And if I did that, I could estimate these beta terms. We're thinking about, let's just say, a case control study. And I'm plotting my true betas coefficients on cases for every one of my 6 categories. So what this is saying is that this first taxon, microbe 1, is much more abundant in cases compared to controls, and taxon 6 is much more abundant in controls compared to cases, and taxa 2 through 5 are a little bit more abundant in controls compared to cases, but not by that much. And so what that identifiability result on the previous slide meant is that we can't distinguish between any vertical translation of these points. So we could shift them all up, we could shift them all down, and on the basis of our W data alone, we can't determine where to peg them. So we have to decide where we want to center them. So one option is to choose a reference category and to say, okay, well, let's say that, let's just set that first category to have a log fold change of 0, and we'll estimate comparisons with respect to that reference category. So when we go to do inference, that sort of very easily, that very easily allows us to ask questions like, is the fold change in one category equal to the fold change in this reference category? But really, we don't care about this reference category. It was arbitrarily chosen. It doesn't seem like a great choice here. So another option that we have is to say, well, we don't care about any one taxon. Let's care about all of them in some measure of center. So we could center by the mean. And so similarly, when we go to do inference and ask formal hypotheses about these betas, we would say, well, actually, none of these fold changes are equal to the mean fold change. And I really like the movie The Incredibles, and so whenever, and I think this is a quote from the movie, but it's like, when every taxon is changing in fold abundance, no taxon is changing in fold abundance. Does that sound familiar? Something like that. I think maybe so the villain who's like, when everybody's incredible, nobody is. It's like trying to take incredibleness away from this family. I strongly recommend. Okay, so what would we really like to do in this scenario? Well, I'd really like to center by something like the median. What's like a robust measure of center for this data? But the median's really hard to work with because it doesn't have continuous derivatives. So when we go to do inference, that makes things really challenging. So what I'm going to propose is centering by some smooth median, something that I'll call smooth median that I'll define on my next slide. And what this allows us to say is, well, the taxa that we would be most interested in, even if we knew what was happening in the real world that we don't get to see, is we're most interested in the taxa that are changing the most in abundance from cases compared to controls. We'd most be interested in taxon 1 and taxon 6. And by centering at some robust measure of center, we can easily see which taxa are changing the most in abundance. Okay, so how are we going to do that? We're going to constrain the rows of our beta matrix using what's called the pseudo-Huber loss minimizer. I'm just going to call it a smooth median. So the way that I like to think about pseudo-Huber loss minimizers is as interpolating between means and medians via some hyperparameter delta. One of the great things about the pseudo-Huber loss minimizers, though, is unlike the median, it has infinitely many continuous derivatives. And so you can think about when this delta parameter is large, our pseudo-Huber loss minimizer behaves like the mean, and when our delta parameter is really small, it behaves like the median. So really what we're coming up with here is this quite easy to work with identifiability constraint that behaves a lot like a median. And if you think about means as, sorry, I should have said this earlier, but if you think about means as minimizers of L2 loss, and you think about medians as minimizers of L1 loss, and you think about the pseudo-Huber loss as moving between the two, I've shown in this bottom panel how different choices of delta allow us to behave more like the mean and behave more like the median. Okay. All right, so now we have talked about identifiability. Let's talk about estimation. Okay, so the model that we've decided to fit is that the log of our expected counts is equal to some sample-specific term plus some regression function, subject to identifiability constraints on the rows of this regression matrix beta, and that constraints arise from this smooth median. Okay, so you might look at this and say, well, that looks a lot like Poisson regression, especially if you're in 515 with me right now. What's happening here is that we've got unknown intensities or offsets. But the way that that's sort of dealt with in this model, if you're thinking sort of more intuitively, is the fact that we've got multiple, we're observing multiple categories for each specific sample. So we are in this case able to estimate those intensities. So the way that we're going to propose to generate estimates of beta and Z is by defining estimating equations using a Poisson likelihood and then maximizing with respect to maximizing that Poisson likelihood. So we're not going to assume that our Wij's are Poisson distributed. We're just using the Poisson distribution as a tool to define an estimating equation. Okay, so we think about these Z parameters as nuisances, and so we can profile them out, do a profile likelihood approach. And what this gives us is constrained multinomial estimating equations. That may or may not have been a Stat 512 homework problem. And so let's think about what happens when we do this. One of the challenges that we're working with in practice is that our count matrices, sorry, I'll pause for questions. Amin, go ahead. Great question. Sorry. So beta is a matrix. I'm thinking about indexing the rows, which denote the different covariates by K. I'm thinking about denoting the columns, which are the categories, by J. And so because of this equivalence class that we can only estimate to within, we need to constrain across the rows of our beta matrix. And so that G pseudo loss is being applied across the rows of that matrix. And we're doing that for every K. That's right. So we've got a constraint on every K equals 1 through 2 rows of beta.
And so what sort of beta? I'm a bit confused. So
let's just think about it as median. We're constraining the median of the rows. We're constraining the median of the rows to be zero. Yeah.
This is not a median. There'll be many beta.
No, we'll actually have only a single element of our, I guess this constrains our equivalence class to give us only a single element, so we can uniquely find a solution to this problem.
Right, but I just focus on the constraint. There are multiple betas.
Oh yeah, absolutely. Outside of the connected estimate. Yeah, for sure. Yeah. Lots of different J-dimensional methods will have smoothly interference. Yeah. No, very good. Okay. Did I think I had a question as well?
Yeah. Minus that notation. Profiling that one lot of Z's. Is that one
and these?
Profiling out one lot of Z's.
Right, yeah. No, it doesn't work. No, it doesn't work.
Yeah, not consistency.
I haven't thought about consistency yet. We're going to think about consistency in terms of our betas, not in terms of our Zs. That's the equation that we like to work in. But yeah, maybe I can come back. But no, not a problem. And that'll show up, I guess. We're going to talk about inference and error rates. So your questions on it in 15 minutes. Let's revisit. Yeah. Other questions? All right. Okay, I'm actually not done with estimation also, which may help. Okay, so a problem that we see a lot in practice is that we fail to observe a lot of taxa, a lot of categories in many samples. Okay, so most of these Wij matrices are sparse. They have a lot of zeros. And so what that means for us is that the maximizers of this objective function that I described on the previous slide may not exist. And so this is a problem that may be familiar to you from, for example, if you've ever been assigned a homework problem looking at logistic regression where you have perfect separation. Okay, so for folks who haven't seen this before, let me talk you through what this looks like in a much simpler example than the one that we're working with. Let's say we're thinking about finding MLEs from a Poisson likelihood, and we're just thinking about Poisson likelihood where we've got our mean as being e to the theta, where theta is our target parameter. So we didn't have a lot of samples. Let's say we had 5 samples, and we had no observations. We had no events occur. So the sum of all of our observations is 5, the mean of our observations is 5. So the score equation that arises from this Poisson likelihood is y bar minus e to the theta equals 0. That's not going to have a solution at any finite theta. Okay, so if we were to think about the population setting here and we were to ask, well, what it would be of the solution to our population score equations, we would have that the true mean minus e to the theta is 0. And maybe it turns out that e to the theta is just pretty small. So it's not too surprising that we didn't observe any counts in practice. Okay, so what this is a manifestation of a broader problem, which is the bias of maximum likelihood estimates in any finite sample setting. And so maximum likelihood estimates have bias of order 1 over n. Where does this bias come from? It comes from curvature in the score. Curvature in the score equation. So you might remember that when we're working in a normal model, we have linear score equations and we also have unbiasedness of MLEs, which is no coincidence. So a solution that David Firth came up with, this very classic paper from 1993, is to adjust the score based on this curvature. So what does this look like in practice? We've got our green line, our sample score, which gets closer and closer to 0, which is what we're trying to solve for. As theta gets more and more negative. We've got this population score that has a finite, valued solution. And it turns out that the Firth correction, this curvature correction, in this case, is adding 1 over 2n to our score equation, and that now has a finite solution, has a solution within a finite parameter range. And so this is a toy example here, but really nice. And I hope it illustrates some of the points that I'd like to make, which is that the penalty that we induce from using a Firth penalty here has 3 really nice properties. Firstly, it's asymptotically negligible, doesn't impact consistency. It's parameterization invariant, so it doesn't matter about the fact that we're choosing this smooth median constraint versus a reference constraint versus a mean constraint. And it also guarantees the existence of an estimator, which is something that my collaborators really like. One of the nice additional aspects of the parameterization invariance is that it has some, makes it easier to do fitting in practice. And I can go into details on that for anyone who's interested. Okay. So the likelihood that we're ending up working with is a penalized likelihood. It's the sum of our profile likelihood and our Firth penalty, which is slightly more complicated in this setting. I won't go into too many more details. One of the really cool things about the way that the Firth penalty simplifies for us is that it allows us to find the maximum penalized likelihood solution here by iteratively fitting Poisson GLMs on augmented data. So the way that this Firth penalty term simplifies allows us to essentially just be left with a Poisson likelihood equation, but on not the W's as our outcome, on some augmented W's. You would think that David would take this as a win and say like, okay, we just augment our data and then we can use GLM, like the R function GLM, to find our MLEs. But nope, that's not fast enough for the scale of problem that we're working with. So we have a super fast Poisson GLM solver using Fisher scoring and a line search. So if you ever need a really fast Poisson GLM solver, feel free to dig it out of the guts of this code. Okay, so the resulting estimator that we end up with is consistent under what I would consider general conditions, general semi-parametric conditions, including correct mean specification. And I know this is an audience largely comprised of, or enriched for folks who think a lot about efficiency, especially in the non-parametric sense. So I will say that this is an estimator that's going to be efficient within the class of models that have correct mean specification and have a linear relationship between their mean and variance, which before I came to UW Biostat, I thought was a pretty broad class of models, and I've since learned otherwise. So I'll say that non-parametric efficient, deriving estimators that are non-parametrically efficient is something that I'm learning about and something that I'm grateful to be working with a couple of our amazing students on. But in particular, I'll note that consistency and efficiency are given to us under more general conditions than Wij being Poisson distributed, which is not something that I would believe to be true in practice. Okay. Ken, did you want to talk any more about consistency? Cool. Awesome. Okay. Let's talk about inference. Okay, so because of this, oh, does anyone else have any questions before I move on? Sorry, I can make special space for Ken, but I can make special space for everyone else too. All right. I'll make time for questions at the end as well. Okay, so we've talked about estimation. Let's talk about inference. So because of this identifiability only to within an equivalence class, we can't test hypotheses about the levels of these regression parameters. We can only test their equality to our constraint. Okay, so what does this amount to asking? This amounts us to asking about differences in the log true abundances across categories and whether or not they're equal, whether they're equal to the smooth median of differences across all categories. Okay, allowing us to highlight taxa that are most overabundant in certain groups compared to others. I feel like this is a nice slide that shows how enmeshed I've become in the intellectual framing of this department, because I was certainly not someone who was very familiar with robust testing before I joined. And now I'm a full-scale convert. So the way that we went about doing inference in this problem was using robust score testing. And I would just say that even for someone broadly on board with this idea of instead of just using WAL tests and score tests, we have robust score tests and robust WAL tests, which is something that many of you will learn about in your applied and theory sequences here at UW. Should we see you in September? But even the point of this is to say that even I was astonished by how well error rate control is maintained for robust score tests. So even in what I think of as the most disadvantageous simulation setting, where we had 250 taxa, zero inflated negative binomial counts, so not Poisson counts, zero inflated negative binomial counts, and 10 samples, we have slight conservatism in our error rate control at 3.8% type 1 error rate, when we're trying to control, we're thinking about a 5% level of significance. That increases to 4.4% type 1 error rate when we have 50 samples, and that approaches nominal as we have more and more samples, of course. So I'll just note, one of the limitations of doing a robust score test is that you have to fit under the null, which is not trivial here because of this complex identifiability constraint. And so we need this more sophisticated augmented Lagrangian algorithm, which I'm not going to talk about today, but happy to answer questions about. So you might say, well, one of the great things that people love about robust WAL tests is that you don't have to fit under the null. You can just use your results under the alternative, but robust WAL tests don't control error rates, especially in small samples. Or sorry, they don't control error rates in small samples. And empirically, we had 30% and 9% error rates for 10 samples and 50 samples. So I'm a realist. I know many of my collaborators spend years taking care of these 10 mice. And so I would say that conservatism is a known property. Is a known property of score tests and robust score tests. And I would argue that it's a good thing, especially in the high throughput genomics setting. So I sleep okay at night with a 3.8% type 1 error rate for small samples, especially in this really disadvantageous scenario. Yeah. Okay, cool. So I'll close with a data analysis before some reflections and open for questions. So the sort of scientific question that we're motivated by in this setting is asking the question of which gut bacteria play a role in colorectal cancer. So Werbel and co-authors in a 2019 study did this incredible meta-analysis where they collected 5 studies from 5 different countries that all had shotgun sequencing data. So this is a maybe slightly more modern high throughput sequencing technique used in microbiome studies, where all of these 5 studies had colorectal cancer cases and non-colorectal cancer controls. So in totality, this was about 550 samples. After we had about 850 categories, and we had have 12 covariates because we're adjusting for study, aka country, whether the sample was collected before or after colonoscopy, the sex of the participant, and then we've got a linear splines for both BMI and age with knots at their median. So nonlinear terms for age and BMI. And the goal here was to identify microbial strains that are enriched or depleted in colorectal cancer samples compared to otherwise similar controls, where we're thinking about otherwise similar as being matched on age, gender, BMI, colonoscopy, and country. Okay. So what I'm showing here are my 845 taxa indexed by the estimated effect size, estimated coefficient on colorectal cancer. And what I'm plotting here in these colors is whether or not these robust score tests were significant at the 10% level after false discovery rate control. So the first thing that you may notice is that I'm plotting robust Wald intervals, and then I'm coloring by robust score tests, which don't coincide. So that's one thing you'll notice here. You'll notice this pretty broad array, and it should surprise you not at all to know that the pseudo-Huber or smooth median of all of these effect sizes is 0, because that's our identifiability constraint. So I plotted that line at 0. One of the things that I really like about this method is that a lot of methods that occupy the same niche in the microbiome literature often really highlight categories that are not very commonly occurring. So those that are, taxa that are, maybe highly significantly differentially abundant, but only present in like 1% of samples. And I like that this method highlights taxa that are present sort of across the proportion of participants in which the taxon is detected spectrum. So if we think about who shows up here, I won't go through this table because I think this isn't the audience for it. But I'll note one thing that was really exciting, which is that the second to largest effect size in the study was a Fusobacterium nucleatum subspecies called Animalis, which was observed at about 24 times expected abundance is about 24 times greater in colorectal cancer cases compared to controls. Fusobacterium is a known player in the colorectal cancer niche. One of the really amazing things about Fusobacterium is that it lives in cancers. So we think about like your skin having a microbiome and your mouth having a microbiome and your gut having a microbiome. And we think in general about your blood as not having a microbiome if you're not septic. It's very dangerous to have bacteria in your blood. And so I just think incredible that cancers have microbiomes, and cancers have microbiomes that are dominated by Fusobacterium. So sort of happening in parallel with collaborators at the Fred Hutch, we were working on this paper about Fusobacterium nucleatum, and I was sort of on the back burner helping out with certain aspects of the analysis and not really seeing the full picture, but that paper's now been accepted to Nature. Its title is A Distinct Fusobacterium nucleatum clade Dominates the Colorectal Cancer Tumor. This work was happening totally in parallel. These results were not connected. And strains isolated from tumors predominantly belong to one of the 4 Fusobacterium nucleatum subspecies Animalis, which is exactly the one that shows up in this table. So there's this nice full circle here. Ali, go ahead. Mm-hmm. Oh yeah, sure. Those colored with the darker color are those that have significant robust score tests at the 10% level after multiple control. Well, yes. pH adjusted score tests less than 10%. True. Yeah. Thanks so much for clarifying. Antonio, go ahead. Oh yeah, and the Q values are the pH adjusted p-values, robust score test p-values. Yeah, thanks for asking. Sorry, so the 2 figures, thanks for everyone for asking. Yeah, so the 2 figures that I showed on this table are all based on the same data. And this paragraph here is sort of a side story related to a collaboration that was happening independently. My apologies, I should have distinguished those 2 things, but yeah. Certainly very satisfying to look at the rank order of this table. All right, okay. So let me wrap up with a summary of what we were talking about today, and I'll close with some comments and some thanks. So the general statistical problem that we're thinking about here is the setting where we've got sample by category tables, and we've got covariate information about our categories, and we'd like to estimate multiplicative differences in means across those categories for every single one of our taxa, for every single one of our categories. Unfortunately, the world is a cruel and nasty place in places other than UW Biostats. And we don't get to see this data. We don't get to see this very nice clean data. We get to see data that's corrupted for every category and also corrupted for every sample. And the question is then, well, can we learn something about this nice clean data from this noisy corrupted data? And the answer is yes, we still can. The only thing that we lose is that instead of being able to estimate the regression parameters that we'd like to target, we can estimate the regression parameters that we'd like to target subject to some identifiability constraint, where we can say we're interested in comparing fold differences compared to typical fold differences, or log fold differences compared to typical log fold differences. So that's what we lose. I would say that this is actually not such a big loss, because when everything's differentially abundant, nothing is. And so we're going to focus on taxa that are the most unusual relative to this smoothed median constraint. Okay. So a really high level takeaway here is that widely used models in genomics and also in ecology are first order misspecified. And I'm talking here about this microbiome example where expected counts, and I talked through this example where if we fail to account for differential detection of the categories in our samples, we can totally be misled, not just about the effect size, but about the direction of effect size. So it may look like that we've decreased the abundance of this pathogen when in fact we've increased it. One of the really amazing tools that we have to understand this phenomenon is control data and mock communities. And I guess I would sort of leave you all with the question, anyone who's working in applied settings, and say, how do you validate the key assumptions that you need in your world? The causal inference folks think about this all the time, but I think genomics folks are thinking about this less often. There's a ton of open work and open problems and ongoing work in this area, and I'm fortunate to work with some just incredible students here. Some projects that I'll highlight include thinking about how do we scale questions like this and methodology like what I've talked about so far in the number of categories that we're thinking about. We're also thinking about how might we support scientists who have far more samples than they're able to sequence, and how can we think about experimental designs that allow them to get the most out of their data. That's with Grant, who's here. I think Sarah's here on Zoom. We're thinking about a number of different data fusion problems where folks are looking at collecting transcriptomic data alongside their taxonomic profiling and so on. That's one problem I'm working on with Shirley Mather, who I believe is on Zoom, as well as a collaborator in oceanography, whose name is Zinka. Ellen's helping me with some think about some of these questions about non-parametric efficiency of these estimators. I think open problems that I would love to collaborate with folks on include how do we, if we're working in this setting where we have some mock community data and then a bunch of samples for which we don't know who's present and in what abundance, how can we learn about differential detection and leverage smoothness across phylogenetic trees? And then there's all these questions about abundances, and I'll just say I work on a lot of different things, some of which don't fit so nicely into this talk today, but in general, my group is interested in any methodological problems that are relevant to biodiversity studies, especially we like those with interesting geometric problems. And I'll also highlight projects by Amir and Maria, who I believe is also on Zoom. Okay, so the bulk of the statistical methodology work that I spoke about today is described in this paper on arXiv called Estimating Fold Changes from Partially Observed Outcomes with Applications in Microbial Metagenomics. As I mentioned, David Clausen has just been the most fantastic collaborator on this project. And I alluded earlier to, we could pursue problem one. We're going to talk about problem two today. Problem one, we talked about in this paper, modeling complex measurement error in microbiome studies. The original characterization of differential detectability of microbial taxa from mock communities was in this paper, Consistent and Correctable Bias in Metagenomic Sequencing Experiments with Mike McLaren and Ben Callahan. And that's been a really high impact paper. So I'm really pleased to see people referring to that more broadly. I think that statistical methodology is only useful and relevant in the applied sciences if it's accompanied by high quality software. And so we make developing open source R package or open source software, generally R packages, a priority. The methodology that I spoke about today is called RADEMU for reasons that I won't talk about. And something that we found to be of independent interest or potentially useful in other settings is robust score testing for just Poisson GLMs, and that's in the R package robust. So I'm open to requests and suggestions. Robust, sorry, because score tests are also called RAW tests. So yeah, robust. We're also very funny and hilarious when we're not developing very rigorous methodology. All of this work is supported by the National Institute for General Medical Sciences, who I'm grateful to have an R35 from. I didn't have anything else to add to the formal part of this talk, but I would welcome your comments and questions. Thank you so much. Thanks so much.
Maybe it's addressed in some of the other papers presented there, but how much of this problem can be solved by just using spike-in? We can do the original sequencing data and you spike it into one of these samples.
Yeah, great question. Yeah, it sounds like we've got some folks here familiar with genomics experimental designs. Yeah, so I am generally a really big fan of spike-ins. The problem is that people analyze the resulting data horribly. And so what people often, so just for everyone else who is here, you know, I talked about how we've got this sort of relative abundance W, and we've got this absolute abundance that we don't get to observe, Yi, that's Yij. And what folks often are doing is saying, okay, well, let's add in to every one, to get around the unknown Zi terms, let's add to every single one of our samples some known concentration of some other bacteria's DNA. And then, okay, we've got some measure of absolute abundance from these samples because we know how much went into it. Let's sort of cross multiply by how much we know was supposed to be in there to try to recover some absolute measure of abundances. And so the problem with that is that it in no way accounts for differential detection across taxa. So there's nothing wrong with the experimental design itself. It's the analytical tools that we're really failing on here. So one of the cool things about this methodology that I spoke about is that if you have spike-in data, you don't need this smooth median identifiability constraint. You can use your spike-in because you know that that's not changing in abundance. So that can become your reference taxon. Yeah, fantastic question. I would be happy to talk about that more. Yeah. Katie.
So in your talk, you covered the differential detection, but at the beginning you mentioned like there's two things you wanted us to see at one point. You might observe some samples where there was zero relative so how did you cover what?
Yeah, fantastic. Yeah, great question. Yeah, absolutely. So I guess for the purposes of the methodology that I spoke about today, I think about the differences between like, you know, zeros and ones as being just sort of typical noise in genomics data. And so that's not a problem for the methodology that we have. I guess I don't think about the need to sort of specifically go out of my way to correct that noise, especially because we're working in the absence of quality control or calibration data in this second project. So I did not specifically address it, but the methodology sort of generally just thinks about noise. And so noise specifically on the presence when something should have been absent, and it's not really special in that regard. One of the things that I'll maybe reference is that the way that people talk about there being problems with sparsity in genomics data in general. So the fact that there's all of these zeros in this matrix Wij is sort of thought to be a problem as opposed to being useful data. And the reason why people think it's a problem is that they like to do log transformations. And how do you take a log transformation of zero? And so I would say that this sort of, you know, for a room full of statisticians who may or may not get to collaborate with folks working in genomics, I would say that this is a really good opportunity to talk about, you know, as soon as you go about transforming your data, as opposed to trying to connect it to some model through parameters, you sort of are losing or certainly losing information about precision, as well as having to make arbitrary decisions about how you're going to handle zeros. So I think there's a lot of room for interdisciplinary communication on like why exactly are zeros a problem, and actually they're a problem because the methodology that people are typically choosing aren't very sensible. That was not the question you asked, but I had a soapbox opportunity. Reception time. Cool. Thanks everyone for coming.